the central nervous system are not topic for today, to this semester. It is for another block, as nervous block in the fourth semester. Because what in the book, in the Jambuila book, explained in the chapter 9 of the book. In the chapter 9 of the book, their part, titled as central nervous system until peripheral nervous system deleted. Today we know about the nervous tissue and also the peripheral nervous system. Peripheral nervous system included the nerve fibers that exit from the brain and also from the spinal cord. As other type of the tissues, the tissue we now about the cells and ECM, extracellular matrix. In the nervous tissue, the extracellular matrix that we know about it in the connective tissue. Yes, this type of ECM not in the nervous tissue. The ECM in the nervous tissue equal to neuroglial cells. Thus, in the nervous tissue, there are just the cells that one type of these cells are principal cells of nervous tissue as these cells that do main function of nervous system and nervous tissue as neurons that conduct and processing and transfer the impulses and responses. And what happened? The ECM in the nervous tissue equal to the neuroglial cells that included some type of the cells that do the supportive regulation protection as same as to ECM that we know about them in this semester. And finally, we now in the peripheral nervous system about the fibers and ganglia. Neuron, main cell of the nervous tissue. Compared to other tissue cells, neuron have a lot of process that other tissue cells, connective tissue, epithelial cells, or muscular cells don't have this process. Each neuron have one part that included the nucleus and organelles named as soma, cell body, or precardium. Precardium. Pre means around, cardio means nucleus. Precardium means around of the nucleus, this part. Soma, cell body, or precardium, that the nucleus and the organelles are in this part. Some short process exit from the soma named as dendrite, and one single process exit from the soma that named axon, that we know one by one about them. Cell body or soma, as same as to other cells, you know in the packaging or some post-translational modification of the proteins that are necessary for the function of the nerve, or no, they should exit from the neuron and transfer to other cells. For the synthesis of the proteins, they send into the mitochondria. They included lots of mitochondria for the function and also for axonal transportation. Lysosomes is another organelle, different type of the lysosome that you know about them, protosome, peroxisome, or others are here. And also these cells, because are long life cells, yes, you know, the neuron are our body for long, as our age, yes, they are long life cells, but you know, they have activity, some materials enter to them by exocytosis, fuse with lysosome, and finally the useful material used by the cell and the waste materials collected in the cell as waste particles. That because these cells, I said for you, the other cell because of short time, for example, neutrophil, just its life is around 2 to 3 days. Then the cell die, yes, the waste also die by itself. But the nerve cell, because of long life, the waste remain in the cytoplasm as our book, yes. We cook some things, yes, and throw all of things to the garbage, yes. They remain. If you do not throw them, yes, our chicken will filled by the waste, yes. In the long life cells occur this one. The waste remain on the cells. That we can see them as hypofashing granules or aging granules. That in the nerve cell and also cardiac cell we can see them. And also some of the normal nerves in the nervous system can store some pigment. For example, the melatonin. Yes? Or there is a structure as substantia nigra that accumulate the serotonin or some hormones inside of them that under microscope they included some pigmented granules. That all of these are the inclusion in the cell. Another organelle are cytoskeleton as neurofilament, neurotubules, intermediate tubules, yes, we know in the cells as microfilament, microtubules, intermediate filaments. In the neuron, we name them the microfilament as neurofilament because they are just in the neurons. The intermediate filaments and the neurotubules are just in the neurons. Neurofilaments included the active filaments, as same as two other cells. There are in the short process, long process for the keeping of the shape of the cell. Microtubules included the tubulin that are more in the axon, involved in the axonal transportation that I explained for you, but and also they included the intermediate filaments that in the cell we have a table that show the intermediate filaments in the neuron. Some of them are in the neuron, some of them are in the glial cells. And about explain about nucleus, finally, the neuron in the soma or cell body included the mist body, purple color, spot, included the RER and ribosomes. And the another spaces that have the white color show the location of the mitochondria, Golgi apparatus, microtubule, or others. Euchromatin nucleus, we use it a lot in this semester and in other semester. Euchromatin nucleus under microscope have white color and show the high activity of the cell in the transcription. Distinct nucleolus and some of the cytoplasmic process. This picture is the Nisle staining mostly for studying in more detail of the nerve cell in the lab. Also, we do the list staging. But in the lab, generally we do the HND, hematoxylin, and eosin that you cannot see these organelles under the microscope. Some short and highly branched process exit from the soma. Branched and short. They are not lined as axon, made as dendrite. All of the organelles except the Golgi apparatus are in the dendrite because they are short and the different proteins or other synthesis don't have to, for example, transferring or other things for a long distance. Because of it, they don't any Golgi apparatus. Maybe it's one of the questions. The dendrite are a site of the reception. They act as a reception in the water. They receive all of the impulses from the internal body or in outer part. Pain, touch, pressure, cold, anything, tasting, smiling, all of the senses, yes, received by the dendrite and then they transfer to the cell body. Schematically, dendrite are the short and branched slides under this. This is a silver staining. It's a special staining for the process of the neurons that you can see the branching of them. And also, again, in each neuron, we can see some spinous-like branching that's named as the dendritic spine. As this. Yes? This is a dendrite. Again, have some as spinous structures. Named the dendritic spine that, as you see here in this picture, they can change their shape and their number. Why? Because the dendrite are the site of the reception. Why? When the impulses are more or no, the our brain or spinal cord active in some skills or some teaching some things, yes, is the increasing or changing of the shape help to us that we able to receive more information. This, the increasing of the number of these dendrite spine and also the changing of the shape finally resulted to more connection between of the cells that help to us for knowing, for the teaching, for we can do some skills that explain for you in the other topic as synaptogenesis. Axon is a one single line process that exits from one pole of the nerves. It has different lengths, maybe around 1 or 2 millimeters until maybe 1 meter or more. Why? Because the soma or cell body just are in the brain and the spinal cord. The soma, pericardium, and the axon exit from the neurons and collected together and finally form a nerve. You know the sciatic nerve also, I see, yes? The sciatic nerve in the lower limb. Optic nerve. Yes, olfactory nerve. Yes, you know them. This nerve derived from the collection of a million axons. Yes, that they soma, for example, the optic nerve soma are in the retina. But the their axon, yes, are long until our fingers. In the tall person, maybe the length of the axon is more than 1 meter because their soma are in the central nervous system. They start from a cone-shaped structure that named the axon hillock. Their axon don't have any RER ribosome or Golgi apparatus. It means the axon not active in the protein synthesis. Anything that the axon need to eat, synthesis in the cell body and transfer to axon. Thus, axon depend in the life of the axon, the activity of the axon dependent to the cell body. And because of it, some person that suffered from the nerve damage, for example, because of the accident or maybe some cutting or others, when the nerve cut, because the axon should receive the nutrient or other things from cell body, they lost in the connection. And in the severe damage, maybe they lost their function. Yes, they don't have any these organs. The cytoplasm that fill the axons, named the axoplasm, and they are surrounded by axolemma. This is just the cell membrane that continue, named as the axolemma. Just their name is different, not their function. The axon hillock from here, the axon lost the RER and ribosome. If we have some session, we can identify it. Some of the axon filaments have a sheet as myelin sheet that form the myelinated fibers, and some of them don't have any myelin sheet. They are unmyelinated fibers. You know, the myelin help to increasing of the speed of the conduction of the impulses or the responses. In the end part, yes, the axon is a long process. That one end of it is axon hillock, that is a continue to the cell body. The other one is a terminal part of the axon. That, as you see here, again have a lot of branching. That's named the terminal branching or terminal arborization. In this part, they connect to next cell and form one structure as synapse. Yes, they have here another connection to next cell. The next one maybe is another nerve, maybe is a muscle cell or secretory cell. For the increasing of the connection, increasing of the synaptic space, they have a lot of branching. And in the end part again, they a little expanded and form a button-shaped structures that because they are in the synapse structures named as synaptic knob or synaptic button. That from here the protein released for the communication to other cells. The axon transmit information away from the soma. The dendrite receive the impulses. Yes? For example, the pain, the terminal part of the axon. This process named as axonal transportation that occur from the soma to terminal part or opposite side. Anterograde transportation from soma to terminal part of the axon. And the opposite direction named the retrograde, come back from the terminal part of the axon to the soma. Which substance or which things transfer? By anterograde transformation, all of the organelles, such as the organelles that are in the axon, such as lysosomes, such as microtubules, microfilaments, and also neurotransmitters that synthesis in the soma transfer from the soma to axon and terminal part of it. As you see here, most of them are the organelles or other proteins. You don't need to memorize this. Just now, in the anterograde transportation, axonal transportation, transferring of the organelles, neurotransmitter, and proteins occur. Which by microtubules and motor proteins. I explained for you in the cell. The microtubules are roads or street. The cars are secretory vesicles. Yes, the secretory vesicles that pass along the microtubules. For this movement, they need to motor. The cars, the vesicles need to motor protein as kinesin and dynein. The kinesin involved in the anterograde transportation, the dynein involved in the retrograde transportation in opposite direction. Maybe it's one of the questions. What things by transfer by retrograde? Some drugs. We eat or inject some drugs, they should go to the nervous system, the other one. They pass from the peripheral to central, peripheral nervous system to central nervous system. Some poisons, as tetanus, as botulism poison, yes, they also transfer by this one. Damaged organelles come back to the ear that removed by the axon. This is the function of the axon that's done in the yes, explain some diseases that in the retrograde one that some toxins or pathogen as tetanus, Clostridium tetanus, or the bacteria can pass around. Now I just explained about the structures of the cells. As same as to other cells, the neurons included the membrane, cytoplasm, nucleus, and organelles. That we know some differences between the neurons to other ones. There are some classification for neurons based on their structure and function. That our topic is just structure. The function is for physiology. Yes, that I don't explain more about it. One classification for type of the neurons is based on the number of the process that exist from the cell body. All of the neurons have one axon. Yes, all of them have just one axon. The number of dendrites are different in them. The multipolar, this is the L, multipolar neurons have more dendrites, more than one dendrite that exist from cell body and have one axon. They involve in the movement. Movement of which? Movement of the muscle. Movement of the secretory cell, glands that secrete some products and exit from them. Bipolar neurons, functionally are sensory neurons, involved in the receiving of some senses as tasting, as vision, hearing, and smell or smell. Smell. E. Yes. Olfactory epithelium. Bipolar neurons have two processes, one dendrite, another axon, that found in the special senses, in the eye, ear, nose, and also on tongue. We have 5 special senses, except the touching. Yes, the 4 other one, neurons are bipolar. This classification also in the exam, especially one of the questions are from the multipolar or bipolar. For example, the bipolar neurons are in which part? Yes? Or for example, the taste buds in the tongue are multipolar, bipolar, or. Yes? Question as this. Unipolar. At first, very short process exit from the cell called soma and immediately divided to process. One acts as an axon, another one acts as a dendrite. These pictures are schematic, yes, but under the microscope we cannot identify them from together as these. Functionally, we can identify that which of them is dendrite, is the reception, which of them is the axon, the transferring part. Where is these neurons? In invertebrate animals, more of their axon is this. In human, in the spinal ganglia and the some nucleus. We know about this ganglion later. This is the classification based on the number of the process that come from the soma. There is another specific classification that just named them that you now name in the physiology as sensory neurons, motor neurons, and associated neurons. Sensory different sense transmit from environment to central nervous system. Motor in the opposite. Send the information to effector organs, muscle, glands, and associate are some neurons that located between of them. That the number of these neurons are more compared to motor or sensory neurons. The type of them is important. The sensory neurons are bipolar. The motor neurons multipolar, and the associated neurons also are multipolar. The another structures, I explained about synapse, then we changing of the materials done by exchanging of some ions. This type named as the electrical synapses, that additional to the CNS, also they are in the muscular tissue, in the cardiac muscle and the isthmus muscle, also there are the electrical synapses. Chemical synapses, two cells, perisynaptic, postsynaptic. Perisynaptic always is a type of neuron. Which part of the neuron? The terminal part of the axon, terminal organization, and also the synaptic nerve or synaptic photon. The end part of the axon, a little expanded, included the secretory vesicles that contain the neurotransmitter. These vesicles exit from the perisynaptic cells by exocytosis, release the neurotransmitter to a space that named the synaptic cleft. Have around 30 nanometer distance between two cells. Neurotransmitter released in this cleft, synaptic cleft. The postsynaptic cells have a receptor for the neurotransmitter. After the joining of the neurotransmitter to the receptors, the ionic channel open and then they do their function. Postsynaptic membrane included the receptors for the neurotransmitter. That is channel open and the exchanging of the ions done between the cells and communicate together and do their function. As this, the nerve cell, here the second one also, or by postsynaptic is a nerve cell, but maybe the postsynaptic be another type of the cell.
The distance is 30 nanometers with the nerve.
Distance, synaptic cleft.
It's 30 nanometers?
Yes. Don't need to memorize, I said for you. Something, for example, the size of the RBCs is important. But the other, because you can compare the size of them to other cells, but most of them not necessary. Electrical synapses. Basis on the existence of the gap junction. Two neighbor cells are neighbor together. There is some narrow channels between that they can change just some ionic molecules by the connection. The connection included the connexin. Here compare two types of the synapse. That in the chemical synapse working based on the releasing of the neurotransmitter. Here there is this distance between the perisynaptic and postsynaptic one. Synaptic cleft, but here two cells are located adjust together and connect together by gap junction. Here there isn't any synaptic cleft. Based on the location of the synapse in the nervous tissue, again there are other types. Which part of the neurons? Yes, connected to other part of the neurons. For example, dendroaxonally, dendrite, synapses with the axon. 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 Dendrite, syn monitor the environment of the neurons, act as a guard, yes, support of the neurons for the entering, exiting of anything, and keep the environment of the neurons in the physiological condition, the pH, the ions, or other things. Some of these processes are close to the capillary of the smallest vesicles in our body. These processes named the perivascular feet. Feet means feet, yes, that are close and around the vessels and involved in the formation of the blood-brain barrier. Is a barrier between the circulatory system and nervous system. Monitor all things that want to enter to the nervous system or the exit one. As I said for you, everywhere that included the barrier, included the tight junction. These tight junctions are between the endothelial cells of the capillary in one side, and in the other side, the perivascular feet of the astrocyte. That finally they located very tightly and very close together, and by the tight junction, they can control the environment and anything that enter to the nervous system. Under microscope, as you see here, they have a star shape, the cell body, and also the process. Also, the astrocyte compared to other glial cells are multifunctional cells. They have lots of function. When we go to the other slide, each glial cell have one specific function. But as astrocyte have lots of function, creating a supportive framework, monitor and regulate the interstitial fluid of the body, creation of the blood-brain barrier. In the damaged part, they can involve in the formation of the scar, and also in the embryonic period, they are important and necessary for the development of the central of the nervous system in the embryo. They have two types. Fibrous astrocyte, protoplasmic astrocyte, that as you see compared to them, you can identify them. The fibrous one have long and few branches, fewer feet and long feet. But the protoplasmic astrocyte have short branch and more branch compared to the fibrous one. Protoplasmic one are in gray matter and fibrous astrocyte are in the white matter. Do you know the gray and white matter? Yes? Yes. Here, for example, in the spinal cord, yes, the gray matter included the soma, cell body. The white matter included the axons. That, because both of these two structures need two supportive cells, the fibrous one are more in the around the axons, and the protoplasmic one are around the nucleus. Oligodendrocyte. Oligo means small. When they discover, at first these cells discovered, and the scientists believe that this part is the smallest one, named it as oligodendrocyte. That is a cell that have lots of process and is small. But now, today, recently discovered another type as microglia that are the smallest one. Oligodendrocyte. For myelin sheath in CNS, they have lots of cytoplasmic process that can, each of these process can wrap around one axon or several axons and form the myelin sheath around it in the central nervous system. Create myelin sheath around the axons in the oligodendrocyte. Compared to the astrocyte have fewer branches. Abundant cells are brain, there are some cavities that you know anatomically about them in nervous tissue. Just now, the central canal of the spinal cord and also the cavities that are in the brain lined by the ependyle cells. They line the ventricle of the brain, this space, and the central canal of the spinal cord. And also, they produce and monitor and circulate the cerebrospinal fluid. In this ventricle, here, and this canal, there is a fluid that have a few amount in the human that its combination is the same as the plasma of the blood, but it's a translucent fluid that circulate in the ventricle and central canal that produced by ependyle cell. As you see here, they have colloidal shape, round nucleus that line these spaces. Microglia. They are a thorny-shaped cells with one soma and some lots of cytoplasmic branch. They are smallest one. They derive from monocyte and form the macrophage of the central nervous system. The oligodendrocyte, the astrocyte, and the ependyle cells derive from neuroblast, from neural tube, but these cells derive from monocyte. Peripheral nervous system included two types of the glial cells, as the Schwann cell or neurolemmocyte and the satellite cell. Satellite cells are supportive cells in the ganglion. Do you know ganglion? The spinal cord included some motor neurons and sensory neurons in the gray matter. That their axon exit from the spinal cord as sensory nerve, motor nerve. Motor neuron, the axon is a motor, sensory are sensory. In the way of the sensory nerves, there is a oval-shaped structures named ganglion. As you know, and I said, the soma or cell body of the neurons are in the central nervous system, in the brain and the spinal cord, central nervous system. But a few amount of them located in the peripheral nervous system. Ganglion. Ganglion is the collection of the soma of the neurons in peripheral nervous system. Yes, the soma of the neurons are in the ganglion. And I said for you, everywhere included nerve cells, included supportive cells. The satellite cells surround the neurons in the ganglion and support the neurons in the ganglion. And also these neurons have another name as ganglionic cells. They are in the ganglion. This is the yellow color is a nerve soma, neuron. That the satellite cells surrounded it as this, supported it. Their function is similar to the astrocyte, provide nutrient, remove metabolic things, monitor the environment of it, and the hemostasis, control the hemostasis condition of the ganglionic cells or neuron in the peripheral nervous system. Here in this small picture show the spinal cord, transverse section of the spinal cord, that the nerve exit from it as this. In the way of the sensory nerves, there is a oval shape. each Schwann cell create sheet for one small piece of the nerve fibers. Create myelin sheet in the PNS. That, as you see here schematically, this is the one axon. This part myelinated by one Schwann, another one. I want to know the difference between the oligodendrocyte and Schwann cell. Both of them create myelin sheet. Oligodendrocyte in CNS, Schwann in PNS. Oligodendrocyte in the same time can myelinate several axons or several pieces of one axon. But the Schwann cell just myelinated a small piece of one axon. It means in the PNS, a lots of Schwann cell can myelinate just one axon. But in the CNS, one oligodendrocyte can myelinate lots of axons. Peripheral nervous system. Yes, until now we know about the nervous tissue as a type of main tissue. Now the neuron structures and different type of the glial cells. You know about the central nervous system in the future, in the 4th semester. That's the part in your book that titled as central nervous system and also meninge is deleted. Then you can continue from the peripheral nervous system. Peripheral nervous system or PNS included nerve fibers that exit from the brain or spinal cord. There are two types of nerve fiber. Yes. Here, as spinal cord. Yes. Included the soma, sensory or motor neuron. Their axon exit and lots of them, 1,000, 100 million of them, located parallelly together and form the nerve fiber as optic nerve, ophthalmic nerve, sciatic nerve, radial nerve, or femoral nerve, or other. This nerve fiber, that is a group of the axons. Yes, some of these axons sheeted by a layer of the myelin. Yes, myelinated fibers. And some of them are unmyelinated. In myelinated fibers, one Schwann cell form the myelin sheet in the one piece of the axon. And again, another one, they myelinated one axon. Between of the part, the piece that myelinated by different Schwann cells, there is a gap that's not myelinated. Why? The myelin sheet included the high amount of the lipid, around 70 to 75% of the lipid, that act as an electrical consultant. It means in this part, not the ions not open. There aren't any ionic channel here. Here, the ionic channel open, they jumped here. The myelin sheet increased the speed of the conduction of the impulses. Axons surrounded by a lipid-rich sheet that produced by Schwann or neurolemocyte cells. That they are very light structures that wrapping of the around the axon. The myelin sheet around the specific of the impulses. For the myelination, that the Schwann cell produces the myelin sheet around the axon, one axon. Yes, that layer by layer wrapped around the axon and finally for the myelin sheet that you see in this animation. The gap between of the part of the nerve axons that myelinated by one Schwann and another one, there is a gap that named the nodes of the run. This part just sheeted but by one layer of the membrane of the Schwann cell. It means in our body, each nerve not bare. All of them at least sheeted by just one layer of the Schwann cell membrane. And also in the unmyelinated axons. As you see here, there are some axons by Schwann cell that just one layer of the Schwann cell sheeted them. In the myelinated fibers, there are lots of layers of the Schwann cell membrane. But in the unmyelinated, there are just one layer that you know. They have a small diameter because the diameter of the myelin sheet and also the speed of the conduction are very, very slow in them. Unmyelinated fibers mostly are in the central nervous system between the interneurons, and myelinated axons and fibers mostly are in the peripheral nervous system for the conduction of the impulses in high speed. There are some connective tissues components in the peripheral nerve as epineuron, perineuron, and endoneurium. What is that? Yes, here, one axon, as you see here. This is Each fascicle surrounded by the peritoneum, this structure. And in each fascicle, we can find lots of axons. Follow my finger, endoneurium, another one. That inside of it, you can find the axon and myelin sheath. Because the myelin sheath is lipid rich when we do tissue processing, these lipids dissolve in alcohol and xylem as adipose, as fat tissue. Because of it, under microscope, we cannot see the myelin because dissolved in the alcohol, right? And just we see the location of the myelin sheath as a white color. But in the practical, I'll show to you. And the last one topic is the ganglion, other part of the peripheral nervous system. It's a collection of the soma in PNS. Based on their function divided to sensory automatic. The sensory ganglion are on the way of the sensory nerves. And the automatic ganglia are in the way of the automatic nerves. Automatic nerves included sympathetic and parasympathetic nerves. Sensory ganglion are the way of the spinal nerves, as you see here. Spinal cord. The gray matter of the spinal cord, sensory neurons, sensory nerve, ganglion, and this here shows the parasympathetic nerve. The ganglion included the soma of the nerves. That the sensory ganglion have a distinct capsule that inside of it there are lots of soma of the neurons, euchromatin nucleus, nucleolus, and the cytoplasm that are spaced in the low magnification, more and more magnification. In high magnification and in more detail, there is a very big cell with euchromatin nucleus and nucleolus. This cell named the ganglionic cells or nerve cells. They are neurons. But because located inside of the ganglion, also name them as ganglion cells. Ganglion cells supported by satellite cells as glial cells. Under microscope, close to the cell body of the cells, there are some round nucleus that show the nucleus of the satellite cells that supported them. The slides that I passed out there, yes, are all in the book, and again, they are not necessary for you. You can study them. If you have questions, you can ask me, but I think you can understand them. But one of the most important subjects in the nervous tissue is the injury of the nerves. As I said for you, the axons don't have any RER or Golgi apparatus or other bond. They depend, their life depends on the soma. When they cut by the trauma, by the accident, by the cutting, pressure, or anything, at first in the acute steps, some changes occur in the neurons. Yes? Here schematically show the normal nerve, the soma, axon. The axon cut or pressured or anything. By this damage or injury, the proximal part of the axon degenerates, degraded, and lost its function. And other changes that occur in the nerve is the chromatolysis. Chromatolysis means the RER and ribosome regress circulation. But in some percent lose out because of the accident, yes, for example, the femoral bone broken, and also pressure to the sciatic nerve. And the sciatic nerve, big part of the sciatic nerves, damaged and distorted.